Okay, good morning, everyone. My name is Shelley Arnott. I'm one of the co-chairs of the meeting, and I'll be introducing and I guess keeping everyone on track this morning. Before we get started, I have a couple of announcements. I just want to remind everyone that tonight there's a poster session starting at 5:30, and for those of you there on Tuesday, you know that it was pretty crowded, and so just maybe a possible tip would be to go a little bit early if you want to try to beat the crowd. If there's a couple of posters you want to in particular see. There's also a few other things going on tonight. The Riverweds documentary is showing at 7:30, and that's here in the Convention Center. And then starting at 8 o'clock until 11, there's a mixer and dance, and that's going to be here in the ballroom. So that's it for my announcements. So the first speaker is Dr. Emma Rosi-Marshall, and she joined the Cary Institute of Ecosystem Studies in 2009 as an associate professor or associate scientist after being on the faculty for 5 years at Loyola University Chicago in the Department of Biology. She earned her PhD in 2002 from the University of Georgia. A stream and river ecologist who focuses on food webs and biogeochemistry, Dr. Rosi-Marshall has published extensively on topics including food webs and heavy metals in urban streams to the effects of genetically engineered corn in agricultural systems. As a result, her work has also spanned a wide geographic range, including such scenic locations as the Grand Canyon and northern Indiana. Dr. Rosi-Marshall is known for her diverse interests in aquatic ecology, her ability to squeeze a 20-minute talk into 15 minutes, and her somewhat controversial areas. A common theme in her research is understanding how anthropogenic compounds affect aquatic ecosystems, which will be the focus of her talk today. So please join me in welcoming Emma.
Thank you all for coming after the late mixer last night and the auction. I appreciate you coming this early. Today I'm going to talk about a role for aquatic ecologists in examining the influence of pharmaceutical compounds on aquatic ecosystems. Before I start, I want to acknowledge my co-author, Dr. Todd Royer from Indiana University. The outline of this talk is I'm going to talk about three main things. I'm going to talk about the pathways of these compounds, how they get into aquatic ecosystems. I'm going to talk about some potential ecological effects of these compounds. And then finally, I'm going to talk about a role for aquatic ecologists and the potential methods that we can apply to this issue. And so we've really tried to outline some things that we can do as aquatic ecologists to address this issue. So pharmaceutical and personal care products, or otherwise known as PPCPs, includes a large range of compounds, things such as antibiotics, antidepressants, anticonvulsants, antimicrobials, hormones, fragrances, insect repellents, and sunscreens. One thing I need to point out about these compounds is they have very different chemical structures, so it's not that they're similar in terms of structure or biological activity, but they are similar in the sense that they're used by humans in our daily lives, and so we group them together because of their source. The other thing about these compounds is that they've all been specifically selected because they have a biological activity. So, you know, we take drugs because they help us get better, right? So they do something in our bodies. So we would predict that these might have effects on an ecological system. And these are just some photos that we've pulled together of various compounds. I do want to point out a couple of interest here. One is these compounds all here, which many of you might use in your daily lives, contain a compound called triclosan, which I'm going to come back to later, and various insecticides and sunscreens. Unfortunately, as an ecosystem ecologist, the first thing I wanted to do is figure out how many pounds or tons are sold on an annual basis or produced. But this information is not publicly available. If someone knows a way to get this, I would be happy to learn about it. But I can tell you that large amounts are used and sold. And so what you can find out, Okay. What you can find out about the pharmaceutical market is that it's a lot of money that is sold. There are a lot of sales. So it's a $773 billion per year market. So it's a lot of money is made in this market, and that's the total world pharmaceutical market. In addition, because these are also, it includes personal care products, and so there are over-the-counter medicines in the U.S., and these are some numbers. These are the top 3 selling over-the-counter medicines. So cough and cold medicines is $4 billion in sales. Analgesics, so painkillers, and toothpaste are $2 and $1 billion in sales. So this is a huge amount of money that is made on these, and we know that we use these in our daily lives. Recently, there's been some work showing that these compounds are detected in surface waters. So a paper was published in 2002 by Dana Kolpin et al. At USGS showing that in streams throughout the U.S., they found a number of pharmaceutical compounds. So 80% of the streams that they sampled, they found, they detected compounds. Now, I do want to point out that they selected their sites based on places where they thought they would find these compounds. So, for example, in the Chattahoochee River, there's a lot of wastewater treatment plants, and so they sampled that area. 82 of the 95 compounds that they were looking for, they detected. And some of the more frequently detected compounds are coprostanol, DEET, caffeine, triclosan, that thing that I told you is in antibacterial soaps. Anything that you use that is antibacterial has triclosan in it. Trichlorophosphate, which is a flame retardant, and 4-nonylphenol, which is a birth control. To the extent that so this paper, Kolpin et al., was cited as the most cited paper in environmental science and toxicology, in ESNT, and it's been cited 1,600 times. But it's only been cited 6 times in JNABS, and it's not been cited, to my knowledge, in L&O. And so to the extent that we view these 2 journals of our societies as representing the mainstream of aquatic ecology, it appears at this time that we are not engaged in this research arena. Okay, first I want to talk about how these compounds get into aquatic ecosystems. There are many pathways to aquatic ecosystems. I've talked to people at this conference, and people say, oh, they're prevalent below wastewater treatment plants. And that is true, but I want to talk to you about some other pathways that they get into aquatic ecosystems. So this is just a diagram of once a compound is manufactured, it's obviously sold, it's either consumed, and some of the compounds are metabolized in our bodies, and then but a majority are excreted, and then they go to wastewater treatment plants, and then some of it gets put into rivers. But there are other pathways as well. So, for example, animal and veterinary uses, where you have a large farm and you don't have a wastewater treatment plant treating the animal waste. The other thing is that we use a lot of these compounds externally, so the antimicrobial soaps, antibacterial soaps, they get used externally and they wash right down the drain. And of course, we know in this audience that sometimes combined sewer overflows will lead to direct inputs into aquatic systems. And then the other thing is not everyone in the world is on a wastewater treatment plant, so there are septic fields where things can get in. The other thing is that people don't really know what to do with unused drugs. And so some pharmacies will have take-back programs, but they can't do it for controlled substances. So if you get a prescription for something like morphine, you can't take it back. And so what you do is you either flush it down the drain or you put it into a landfill. And so then the pathway might exist that way. The other thing is that these compounds, when they get into wastewater treatment plants, depending on their chemical structure, they may sorb to activated sludge. And so, for those of you who don't know, depending on where you are, we take this activated sludge and we either apply it to agricultural fields as a fertilizer, it's called Biosolids, and so that's a pathway into aquatic agricultural systems. And then also, we use that activated sludge in our landfills. So we put our trash, and then we put biosolids in trash, and biosolids like a layer cake. So there are many pathways of these to aquatic ecosystems. We know that these compounds are found in rivers, and I'm not going to go through all the literature on what's found and where it's found, but this has been a global effort. A lot of people have measured these compounds in aquatic ecosystems throughout the world, so that's well known. It's also well known that this pathway of wastewater treatment plants is a dominant pathway, but it's less well known how much gets in from combined sewer overflows or septic fields or in agricultural areas. Okay, so in terms of PPC removal during wastewater treatment, this is variable. It's compound specific. So if you go to a wastewater treatment plant, you're not going to find everything that people use because, as I told you, these compounds differ in terms of their chemical structure. So they differ in their absorption capacity. They vary in their susceptibility to breakdown, whether or not it's microbial or photodegradation. And then, of course, not all wastewater treatment plants are created equal. Different plants have different types of processing. So if you go to a river below a wastewater treatment plant, you may find a suite of compounds, and that reflects what occurred during the wastewater process. To show you about the sludge that's applied on agricultural fields, or the biosolids, we do this in the agricultural Midwest. We take sludge from large wastewater treatment plants and we extract the water and we apply it as biosolids. And this is a paper that was published in 2009. And just I want to point out to you that these are the various compounds, some that I mentioned, things like triclosan, ibuprofen. I'm going to talk about carbamazepine later. These compounds are present in the biosolids in relatively high concentrations, and this is in the pore water, as that after you apply that biosolids to the land, these compounds leach out over relatively long, this is the duration of the study, over 36 days. So once we apply those biosolids to the land, these compounds can then leach out and get into agricultural streams. Septic systems also don't remove all of these compounds. This is an example from a campground in Canada that serves only 2,000 visitors annually. And what you can see here, so this is the drain field from a septic, and you can see this plume of ibuprofen in the groundwater and chlorfibric acid. And you look at those concentrations, 1,500 nanograms per liter. That's not that small of a concentration, and this is just for 2,000 visitors. So I'm hoping that this is dispelling the idea that it only is below wastewater treatment plants. The other thing is that, we have in this country and in the world a lot of leaky infrastructure, right? So our wastes go in pipes underground and get delivered to the wastewater treatment plant. But in an area that Todd Royer, my co-author, studied, what he demonstrated by taking samples above and then below the wastewater treatment plant, that during precip events, so during rain, 70% of the pharmaceutical load can be coming from non-point sources. So above any wastewater treatment plant, he was finding concentrations of these compounds. So I hope I've demonstrated to you that these compounds are ubiquitous in aquatic ecosystems. It's a common myth that these only occur below wastewater treatment plants, and this is not the case. And so what I argue is that ecological studies may be occurring, the work that all of us do on rivers and lakes may be occurring in a sea of PPCPs. So just because you haven't measured them doesn't mean that they're not there. And that the ecological phenomenon that maybe we can't explain, some variability in a study, may be due to the presence of these compounds. Now this is totally speculative, but it may be the case that these compounds influence the ecology of our study sites. The current research on pharmaceutical compounds is mainly in three areas. Analytical chemistry, so how do we measure these compounds effectively? Fate studies, do they get into aquatic systems? And then toxicological studies, at what concentrations pose a threat? And what we argue in this talk is that we need to really join the fray. And as aquatic ecologists, we need to join this issue to answer the question, what are the ecological effects of these compounds? So just to give you a little illustration of some of the potential ecosystem effects, I want to go through some of the literature that's come out recently that you may not be aware of. There's a paper that came out in 2010 showing that plants are affected by Cipro. So Cipro is a common antibiotic. Probably everyone in here has taken it at one time or another. And we don't often think about antibiotics affecting plants, but in this case it reduced photosynthetic activity, this antibiotic. Some recent research that Todd and I have been conducting, we've demonstrated that diphenhydramine, which is Benadryl, so an very commonly used antihistamine, suppress the growth of algae relative to control. If you look in the literature and you look for effects of Benadryl on algae, I can tell you there's nothing out there on it. So another compound, this really widely used compound called triclosan, it's in about 96% of the soaps that we use. It's really common. There was a nationwide survey conducted, and they showed that pretty much no one that they found in the United States did not have triclosan in their urine. And they looked at children all the way through adults. So it's a very commonly used compound. What we found is that below a wastewater treatment plant and in a suburban stream, we found that there's increased resistance to triclosan in the microbial community. And so what is that doing to these microbial communities and processes? A recent paper came out showing that when leaves were exposed to an antibiotic mixture, you basically suppressed the bacterial growth and promoted fungal growth, which influenced the selection by shredders for those tissues, And so, you think about a system that has antibiotics and how that might influence the shredder fungal relationships. In terms of invertebrates, there's some recent data that we've collected in my lab showing that an antihistamine, and I'll talk about this a little later, reduce the growth and production of an invertebrate. And so we don't often think about antihistamines doing this, but I'll explain a little bit later why. Carbamazepine, which is a very common anticonvulsant, so it's used to treat epilepsy, but it's also been used to treat bipolar and helps people with alcoholism. And it's commonly found in aquatic systems because it's really not broken down or reduced through wastewater treatment plants. And there was a study that was published in 2005 showing that when carbamazepine is in high concentrations in the sediments, the insects didn't pupate and emerge. So it stopped them from completing their life cycles. Also, a recent paper was done that showed that anti-inflammatories and propanol, which is a beta blocker, in combination with temperature, explains 71% of the taxonomic variance in an invertebrate community. And finally, this same compound, carbamazepine, this anticonvulsant, in rainbow trout causes brain damage to the trout. And so this is commonly found in rivers, and it also results in liver damage. So we think about streams and aquatic ecosystems, and this is this is what looks like a really pristine system. It's right on the Cary grounds, and in fact, it has concentrations that are detectable of carbamazepine, which is that anticonvulsant. It has ibuprofen and acetaminophen in the water. And so for the rest of the talk, we're going to talk about how might our approaches and methods and perspectives contribute to this emerging global issue. And I pose the question, do we have to develop all new techniques, or can we apply our existing methods to this question of PPCs in aquatic ecosystems? And so what I'm going to do for the remainder of my time is I'm going to talk about how we can take common aquatic ecological methods and apply them to the question of pharmaceuticals. So first, we study solutes and particles in aquatic ecosystems. Stream ecologists have very well-developed methods for measuring solute dynamics and particle dynamics. Both of these are important because pharmaceuticals can sometimes sorb to organic matter and be in the dissolved phase. So we have well-developed methods for measuring things like ammonium as it moves downstream, and we argue that we could use these same methods to look at pharmaceuticals. We could take advantage of existing sources or as drips, so downstream of a wastewater treatment plant. The other thing that we could do is we can do controlled releases of pharmaceuticals. Obviously we wouldn't use an IV bag, but that we can drip pharmaceuticals into stream systems and look at how far they move and their transport distances. And this offers an excellent collaborative opportunity to work with analytical chemists. I won't lie to you, the chemistry to measure these compounds is very expensive and difficult, but this is constantly changing. New methods are rapidly emerging to look at pharmaceutical compounds in river systems, and so the literature is advancing quite quickly. And so we argue that spiraling provides a conceptual framework that is applicable to these compounds. The other thing is that we have biogeochemical assays well developed in our field to look at things like nitrification or denitrification. And Gene Likens wrote a paper a number of years ago saying that, you know, these biologically active compounds, he was talking about PPCPs, probably have major direct and indirect impacts on biogeochemical cycles. So we can measure biogeochemical processes using assays. So if we amend these assays with compounds such as PPCPs and measure how they influence biogeochemical processes, we could really advance our understanding. So just to give you an example, we found that below a wastewater treatment plant in a suburban system, which is this right here, this is a suburban, no large wastewater treatment plants above that site, we found that a large proportion of the microbial community was resistant to this compound. And of course, I don't need to tell you all that all microbes are not the same. They have different functional properties, and what they do in terms of biogeochemistry is different. And so are we selecting for certain members of the community? Is it all microbes that have resistance? And so this could be a fruitful line of research. The other question is, can we use diffusing substrates? So this is, we commonly use, people often are familiar with the flower pot method, where you take agar and you amend it with nutrients and it diffuses slowly out through some kind of a flower pot or something. And so we commonly use this method to look at nutrients, but we've modified this method so that we now have pharmaceutical diffusing substrates. So you can amend the agar instead of with ammonium and nitrate. We can amend the agar with things like caffeine or Benadryl, for example. This works only, obviously, with soluble compounds. Not all of these compounds are very soluble. And so we use the same methods, but we put a lot lower concentrations in to mimic the fact that the pharmaceuticals are found in much lower concentrations than nutrients in streams. And this allows you to examine whether important processes are influenced by these compounds out in the real world. And so do these compounds affect algal or microbial growth rates, right? And what we found was we added a number of different compounds to our little cups. So our agar's in there, and the pharmaceuticals then diffuse out through a substrate here. And what we found, this is the control. This is ranitidine, which is another antihistamine, and this obviously caffeine that we all are on right now. Diphenhydramine is that Benadryl. And what we found is that gross primary production was significantly reduced relative to the control when these biofilms were exposed to these compounds. And we conducted this both in Indiana and New York, and we got consistent results in both places. And also we had a reduction in chlorophyll A as well. The other method that we could use is we have well-developed methods for using limno corrals and artificial streams or streamside channels. And so we did an experiment looking at the effects of cimetidine. Cimetidine is Tagamet. It's used for acid reflux disorder. So when you have, high amounts of heartburn, you can take cimetidine or Tagamet. The interesting thing about Tagamet, it was the first drug to reach 1 billion in sales. So this is a very commonly used drug. The thing that I want to tell you, though, is that invertebrates use histamines as neurotransmitters. So they use histamines very differently than we use histamines. It's a neurotransmitter, and we discovered this by reading literature that was way outside of aquatic ecology, so neurophysiological literature. We found out that if invertebrates were exposed to cimetidine, their pyloric valve function slowed way down, and that was the neurophysiologists were trying to figure out what they used to control the pyloric valve function. But of course, I came along and said, well, if these invertebrates are exposed to this, how might they survive differently? And so what we did in these artificial streams is we did an 82-day experiment. We added cimetidine to these streams at the concentrations that were reported in the Kolpin paper. So these are not, 1,000 times normal. These are the concentrations that you find. And what we found is that there was a reduction in secondary production of this invertebrate in the artificial streams when they were exposed to this compound at natural concentrations, or at concentrations that are found in the field. So obviously, I don't think I need to explain to this audience that, the implications of this are pretty great. So if we are reducing secondary production in these areas as a result of these compounds, this could be really important for food web dynamics, et cetera. We also can use whole system experiments. These are powerful tests of looking at various compounds. We've done work in the past adding nutrients to lakes or adding nutrients to streams, and you can do a long-term addition of these compounds. And this would be an excellent way to examine the effects of these compounds in aquatic ecosystems. To my knowledge, there's only one of these experiments that's been done. This was published in PNAS a couple of years ago. They added, in this experiment at the experimental lakes area, Kidd et al. Added 17-ethinyl estradiol, which is the birth control drug. They added it for a number of years, and what they found was that the fish on the fathead minnows became feminized, and the population crashed. And this is at concentrations that are measured in the field. These are not very high concentrations. And so, you can imagine the implications of this for aquatic ecosystems. This is very speculative. I'm not sure that this would work because I haven't found any evidence of anyone doing this, but we could use isotopic tracers to look at how these compounds move through food webs. So Balmer et al., again, this triclosan, this generalized antimicrobial, with increasing population in a watershed, they saw increases in the concentration of triclosan in the tissues of fish. So triclosan, I told you, is a generalized antimicrobial. It also has androgenic compounds. So it actually, in humans, it's thought to reduce sperm quality and actually it acts like testosterone, and it increases virility, but reduces the sperm quality in humans. And so we don't know what it might do in fish. But we can buy, if you do a search on the internet, you can buy isotopically labeled PPCPs. You can buy tritiated ones, you can buy deuterated ones, or ones that are labeled with 13C. So we could apply methods that we've already developed using mixing models to look at these pharmaceuticals through food webs, because it's very complicated and expensive to actually measure the real concentration in organisms. And so this may be a powerful tool that we could apply. So in summary, I hope I've convinced you that PPCPs are commonly found in aquatic ecosystems. It's not just below wastewater treatment plants. If you have humans in your watershed, you probably have pharmaceuticals in the ecosystem that you're studying. I mean, it's only probably in the very remote headwaters of places like Coweta, but if there are people using drugs, which people like to do, then they are going to be in your aquatic ecosystem of interest. And just because you haven't measured triclosan concentrations or carbamazepine doesn't mean they're not there. And I hope I've convinced you that they might have ecological effects that might influence the systems that you're studying. And what I hope I've also convinced you of is that we don't have to become toxicologists to look at this issue. We can take methods that we've already applied, that we have well-developed methods in measuring ecosystem functions, to look at the ecological effects of these compounds. Right? And so we can really add, the toxicology is important, but the ecosystem function and how organisms are interacting in aquatic ecosystems is also really important. And this group here, have the tools to measure that. And so I think we should maybe get to work on this. So in conclusion, I think that aquatic ecologists need to play a larger role in this issue. Our methods and perspectives really do provide a unique and valuable information. I can tell you I've read a lot of these papers. They don't measure things like, the V sub F of ibuprofen in a river system. For those of you who study streams spiraling, you know what I'm talking about. They don't measure gross primary production, right? They don't measure invertebrate secondary production. These are things that we are very good at doing, and that when these compounds get into aquatic ecosystems, they may influence important processes that we are interested in. And so basically we argue that ecological information can play a role in managing these compounds, but only if aquatic ecologists are engaged in the issue. And so I really hope that we've convinced you that the methods are already in place. We can get out there and do some work on these compounds. I think that if we want these compounds to be considered and their ecological effects to be considered when they're potentially regulated in the future, which I should point out, they're not regulated now, that in the future, if we want ecological information to be included in our evaluation of these compounds, then we need to do the work. We as aquatic ecologists need to engage this issue. And with that, thank you for your attention.